Let's talk about the potential effects if there were defects in photosynthesis and the Calvin cycle. So first of all, what if Photosystem II cannot perform its function? What would we expect to happen? First, we would still expect water to break apart and donate electrons, but Photosystem II would not donate the electrons to the next protein in the channel. So electrons would not flow through the electron transport chain, which means no hydrogen ions will be pumped from the stroma to the lumen. This means that no electrochemical gradient is created, and as a result, no NADPH is formed. Because in order for NADPH to be formed, NADP+ must accept electrons, but NADP+ will have no electrons to accept. Also, no ATP is formed because hydrogen ions will not flow through the ATP synthase, so no energy is created to fix ADP and Pi into ATP. As a result, no NADPH and ATP are produced, and NADP+ and ADP will accumulate. Let's do another example. What if there was an issue in the cytochrome complex? So what if the cytochrome complex is unable to accept electrons? Well, if we block cytochrome complex, then PQ will still hold its electrons. PQ will be unable to give its electrons to cytochrome complex, and this means that all proteins before cytochrome complex will have electrons, but the proteins after will not have any electrons. This means that everything before the complex will be reduced, and everything after the cytochrome complex will be oxidized. Now let's do another example. So what if we blocked Photosystem I? What if there was a defect in Photosystem I? So if there was a defect in Photosystem I, then we would not make NADPH. This means that the Calvin cycle would stop because NADPH is a reactant in the Calvin cycle. So we will eventually stop making glucose in the Calvin cycle. And for another example, what would happen to ATP concentration? If we made our thylakoid membrane permeable to protons. So first of all, this is our thylakoid membrane. And if we made it freely permeable to protons, then our H+ ions would be able to move through the thylakoid membrane without requiring the energy that is released from the flow of electrons. So our electrochemical gradient of H+ ions in the thylakoid lumen would decrease, since H+ ions will flow back into the stroma. This means that we will have less H plus ions flowing through our ATP synthase protein, so less ATP is produced. What about NADPH concentration? What would happen to our NADPH concentration if we made our thylakoid membrane freely permeable to protons? So if we allow H plus ions to flow freely through the membrane, then less ATP will be produced. This is because less H plus ions will flow through the ATP synthase. However, NADP plus will still be reduced to NADPH. This is because electrons will flow through the ETC. So we have a lot of NADPH, but we don't have ATP. This means that we cannot do the Calvin cycle because the reactants of the Calvin cycle are both NADPH and ATP. You need both of them to do the cycle. You don't have ATP, so NADPH will accumulate.